And this is an interdisciplinary research institute with special focus in frontier themes in bioscience. So with this, I just would like to welcome and thank you very much for your time, for being with us today.
So
I cannot hear.
Dr. Roy, I'm sorry, we cannot hear you. Should we just test the sound a little bit?
can you hear me now?
Yes, much better.
Okay,
Thank you.
Okay, great. Hey, I'd like to talk. Thank you very much for the kind introduction. Fernando, thank you for inviting me and setting this up. I'm happy to be here. What I want to do today is give you talk about some of our more recent work. And I want to make two main points. First, I want to talk about the role of RNA, forming RNA interactions to promote the formation of these large RNP granules that we're all familiar with in cells. And I want to emphasize that we've discovered that the really abundant dead box protein EIF4A plays a role in keeping apart these RNA-RNA interactions in order to limit RNA-RNA aggregation. And this makes me start to think about RNA assembly and RNP granules similar in some ways to protein aggregation in disease. And the implication of that made us think that, maybe some of these protein aggregation diseases are really RNA protein aggregation diseases. And if I have enough time, I'll talk about some of our work on tau aggregates, where we show that tau, which is connected to many neurodegenerative diseases, is full of RNA in their assemblies and has some interesting properties similar to RNP granules. Okay, so that's my last slide. So the way I want to get there is by reminding you all that RNP granules are really ubiquitous in eukaryotic cells. There are a diversity of these different ones. There are things in the cytoplasm like stress granules, and also many nuclear RNP granules such as perispeckles. And there are some very interesting cell type specific RNP granules such as neuronal granules and also maternal RNA granules that we'll hear a little bit about from Liz later. Now, I've been very interested in these, I think, for really, three big reasons. One is that RNP granules do some very interesting biological roles. And in this slide here, what I'm showing is some experiments we did with Mani Ramaswamy. And the important take home from this is that if you disrupt the ability of Drosophila neurons to make these RNP granules found in neurons, they're unable to remodel their neurons in response to training. So they're actually defective in making memories. And so RNP granules are really interesting in that they affect synaptic plasticity, they affect stress survival, they affect the localization of RNA in a variety number of contexts. We're also interested in these because they contribute to a number of different degenerative diseases. I think one of the things we've learned in that there's many mutations that the mutations can actually be causative in disease, and that those mutations tend to make the RNP granules more static or more forming. If you reduce RNP granule, you can suppress that neurotoxicity in some contexts. And then finally, I think RNP granules are also interesting model systems for trying to understand how cells build large assemblies and what they can do with them. Now, in my lab, we often use stress granules as a model system for studying RNP granules. Stress granules are terrific in that they form during a stress response, and we can rapidly induce them or trigger their disassembly. And so what I'm showing you here is, in a normal cell, most mRNAs are engaged with ribosomes, and they're translating. And in this picture here, red dots are specific mRNA detected by single molecule FISH. And that when you trigger a stress response, which inhibits translation initiation, ribosomes run off, mRNPs collapse into a relatively tightly folded structure, and then those RNPs, some of them will condense into a stress granule, which we see as the large green dots. We know a lot about stress granules from my work and work of other labs. They largely form from untranslating RNPs. They have a very complex proteome, which is dominated by RNA binding proteins, and they're quite dynamic. And we didn't really understand their function. And so one of the things that we were interested in a few years ago was trying to understand what RNAs are actually in stress granules. And three members of my group, Anthony Kong, Tyler Matheny, and Samya Jain, had worked out methods to purify stress granules, and then they purified them and sequenced the RNAs. And the surprising thing that we found was that there wasn't a real clear distinction of what RNAs were in granules. And I'll come back to this at the end if there's time. But what there really was a very strong bias towards long RNAs. The average RNA in a million stress granules is about 7 kb, whereas the average mRNA in a million cells is about 3. So there's also effects of translation efficiency and some sequence elements, but the most important metric was length. And so why are long RNAs in stress granules? And we initially thought the simplest model would be that if you have a long RNA, when ribosomes run off, you'd expose all these RNA binding surfaces for proteins, which would target you to stress granules. And however, we realized that we couldn't figure out where those RNA binding proteins would come from, because you'd have to have a very large pool of RNA binding proteins not engaged with RNA in the cell. And we really didn't see much correlation with whether an RNA was in stress granules and whether it had binding sites for proteins known to go to stress granules. So this suggested a second model, which really is that when ribosomes run off, you expose a lot of naked RNA. And that naked RNA, of course, can form RNA-RNA interactions with other RNA molecules, and that might actually drive the assembly of stress granules by RNA-RNA interactions and trans between individual mRNPs. And at the time, Brianna Van Treek, who was a grad student in the lab, did kind of an outrageous experiment where she said, if that's true, then we should be able to reconstitute a stress granule without any proteins. And she basically took either yeast or mammalian RNA, put it at the concentrations that we estimated would occur in the cytosol when ribosomes would run off of mRNAs, and asked what would happen. And what happens is those RNAs coalesce into these little RNA particles. There's no protein here. This is just RNA self-assembling under roughly physiological conditions. And kind of the striking thing about these was if we purified and sequenced them, we found that they largely overlapped with the RNAs that we already knew were in stress granules, either in yeast or mammals. So in other words, the biophysical properties of the RNA that are driving self-assembly in vitro actually correlate with the features that drive RNAs into stress granules in vivo. And this suggested to us that, that stress granules assemble in part through RNA-RNA interactions. And once we said this, we realized, oh, of course, right? And explained a lot of other observations in the literature. If you inject RNA, Erica, thank you. If you inject RNA cytosol, it will actually form stress granules in a wide number of contexts. If you increase the intracellular salt in cells, you'll actually promote stress granule formation, which will stabilize RNA-RNA interactions. And if you decrease the intracellular salt, which decreases RNA-RNA interactions, you'll disassemble stress granules. So we think that RNA-RNA interactions play a part of the role in assembling stress granules. But I want to emphasize that they're not the only contributor. There's been a large number of really decades of experiments identifying proteins that can affect the ability of RNAs to form stress granules. And this is just a classic one, probably one of the most important proteins here, is this protein G3BP, which if you delete it from cells, it can no longer form stress granules. And we can understand how G3BP works because it's an RNA binding protein that can also dimerize, and it can link RNAs together to make a higher order assembly. So proteins can contribute to this assembly as well. And so the way we think about RNP granule assembly, starting from stress granules, but I think we should extrapolate this to essentially any other RNP granule in the cell, is that the ability to form a granule is really a summation of two kinds of interactions. One would be protein-protein interactions of RNA binding proteins that are stuck to individual mRNPs. And the other would be interactions between RNA-RNA molecules in trans. And that in a normal cell, you would start out here with no stress granules. But when RNAs are released from translation, you'd expose all this RNA. They would form stress granules as you shift across this assembly diagram. If you get rid of the key protein that drives their assembly, you move down this protein-protein interaction and you get to a place where there's no stress granules. And so this, I think, illustrates you can actually make very protein-dominated assemblies of RNP granules, or you can make very RNA-dominated assemblies. And in many cases, it's really the summation. Now, since many years, people have been working on this axis. In my lab, we decided we'd focus on what are these RNA-RNA interactions which promote stress granule assembly. And two members of my lab, Devin Tauber and Gabe Tauber, decided to work on this. And I want to just make a few general points about RNA in terms of driving assembly of granules before I go on. The first is that RNA is really good at self-assembly. We tend to think that many of you have seen experiments where people have done protein self-assembly, either by liquid-liquid phase separation or by other kinds of self-assembly reactions. But RNA, many of these experiments are done in the order of mgs per mil, whereas RNA self-assembly happens under very physiological conditions and at concentrations of RNA, 150 per mil, that's relatively physiological. We can even make RNA self-assemblies at concentrations as low as 2 micrograms per mil. So I would say to you, any of you who are working with purified RNA in a test tube, if you have any salt in there and it's a long RNA, you probably already have RNA oligomers at least forming in your test tube. So not only is RNA good at this, it's any RNA. We can make self-assemblies with any homopolymer because of the diversity of interactions between the bases in RNA. And we can do this with essentially any RNA that we try to. So it's not limited to specific classes. Any RNA can do this, although some are certainly better than others. The final point I want to make is that if you make an RNA assembly or you make an RNA protein granule, it will recruit other RNAs to its surface because it has RNA molecules on its surface as well, and those will tend to interact with other RNA molecules. And so this is an example of this where we have a purified stress granule here, tagged with a fluorescent marker. And if we add an exogenous fluorescent RNA, you can see it's recruited to the surface of that granule. You can also do this with the assembly of RNA droplet just made out of poly-A RNA. And you can see a particular RNA in green here is recruited to its surface. Now, the reason that's interesting to us is it suggests a real problem that we call an RNA entanglement catastrophe. So imagine you have an RNA granule in a cell, and you've got RNAs in solution floating around. They're going to want to absorb to this surface on the RNA granule. And you create a high level concentration of RNA at that site, and that those RNA molecules will then tend to interact with each other and form an even more stable assembly going down into a lower energy well. And we can actually show this occurs in cells in vitro. Here's a case where we made an assembly with a poly-A core, and then we added an RNA to it. And what you can see is if we dilute that RNA, that mixture into water, which destabilizes RNA-RNA interactions, you can see that core goes away and the surface persists. This is what people would call a Pickering emulsion in the surface chemistry business. And those structures persist because we can actually show that the RNA molecules have actually formed small duplexes between each other, which are stabilizing their interactions. So this creates a conundrum for us, right? Is that, for 10 years, we were trying to understand how cells make RNP granules. And now we're trying to understand how they avoid making RNP granules because RNA is going to be really good at this when it's not engaged in translation. And so what we think then is that cells contain mechanisms which actually keep RNPs apart. so that you not only have things which can promote assembly, such as longer length or high level concentration, but then you're going to have things which push RNAs from assembling these larger granules to being more monomeric. And that would be things like ribosomes would play a big role because they would keep the RNA engaged in a different state. But we also focused on the potential role of RNA helicases in taking apart these structures. And Devin, when he began his PhD in my lab, really wanted to ask the question of sort of what helicase might be involved in limiting the formation of RNP granules in cells. And to do this, the first thing he did was he asked, well, what helicase could be involved? And so this figure shows a number of different RNA helicases which are found in stress granules and their relative abundance in the cell. And you can see there's a lot of helicases involved associated with stress granules, but you can see this one out here is an extremely abundant protein. This is the protein EIF4A1. It's been studied for decades as a protein which interacts with the cap binding complex to help bring in ribosomes to promote translation initiation. Now, it's always been a mystery in the field as to why this protein is 10 times more abundant than the cap binding complex. And there's so much of this protein in the cell, there's on the order of 10 to 15 molecules of this for every mRNA. So why do you need so much of this protein in the cell? And so we hypothesized then that this protein is actually taking apart these RNA-RNA interactions and limiting that formation of RNP granules. So to test this, we did a number of different experiments. The first one I want to show you is a very simple one. It goes back to our way we think about how RNP granules assemble. You know, normally what we would say it happens in a normal cell, there are no granules. When you release mRNAs from ribosomes during a stress response, those RNAs now increase the number of RNA-RNA interactions, and you move across this axis to forming assemblies. But the cell doesn't particularly want that to happen, so it has these RNA helicases, such as potentially EIF4A, pushing you back this way by taking apart these interactions. And so you end up here and you see granules assemble. Now, if we get rid of a key protein like G3DP, we come down here, we don't see any stress granules because we've limited the protein-protein interactions. But a prediction would be we could restore those granules if we now inhibit 4A and we allow RNA actions to increase again. So this is what that experiment looks like. This is a wild type cell here. And here we're doing oxidative stress. You can see stress granules are forming. If we get rid of this protein G3PP down here, they do not form. Now, if we add a drug called hyperistinol, and what hyperistinol does is prevent 4A from binding to RNA. It doesn't make any difference in the wild type cell, but in the G3AP mutant, you can see that you now form these small stress granules. If we add a different drug, patamine A inhibits the ability of 4A to bind to the cap binding complex and promote translation. If we block that interaction, it doesn't restore because EIF4A can still function as an RNA helicase under those conditions. And I just want you to notice, you know, these stress granules are smaller than these. And that made us think, well, maybe there are other ATPases that are playing similar roles. And so if we deplete ATP, you can see that we restore stress granules almost as much as in the wild type cell. So what this suggests is that 4A is playing a role to keep RNAs apart and preventing stress granule formation. We can do the experiment the other way around. For example, a prediction of this would be if we overexpress 4A, we would now move further to the left on this axis of RNA-RNA interactions. And that might take a cell that normally makes stress granules to no longer making stress granules. And that's what you see over here, whereas we're overexpressing the EIF4A protein, and it's tagged in green so you can see which cells. And you can see that this cell here, which overexpresses 4A under a stress condition, does not make stress granules, where its neighbor here that was not transfected does make stress granules. So EIF4 not only, it can limit RNA granules when it's overexpressed. All right, so how does it do this, right? So, you know, many of you have read about dead box proteins, and dead box proteins are referred to as RNA helicases. And I want to point out, I think this is really a misnomer, and that we should call these proteins not helicases, but ATP-dependent RNA binding proteins. And the way these proteins work is they bind ATP, and in the presence of ATP, they can bind to duplexes and induce a bulge and transiently dissociate and reduce the stability of that duplex. If that is a short duplex, then the strands can separate, and then for the protein will eventually hydrolyze ATP, and when it hydrolyzes ATP, it's released from the RNA. If this is a really long duplex, then this will bind, it'll hydrolyze ATP before the duplex is disrupted, and you'll end up with this futile cycle. And so what I want to point out then is this type of helicase is not designed to unwind large duplexes. It's designed to destabilize short, possibly imperfect duplexes that might form in a very promiscuous manner between random RNA surfaces that are not engaged with other proteins or ribosomes. And so our prediction then would be that the way that 4A is limiting this RNA self-assembly is not by acting as an ATPase, but it's just acting as an ATP-dependent RNA binding protein. And that if we could test this, we would see that all you need to do is be able to bind ATP and RNA, and you would be able to disrupt granule formation. And so we tested this using the overexpression experiment. I've already showed you that if you overexpress EIF4A, we can block stress granule assembly. So then we're going to overexpress a variant of EIF4 that can no longer bind RNA, and also a variant that can no longer hydrolyze ATP. And if you do that, what you see is that here's the cell overexpressing wild type, and it no longer makes stress granules. Here's a cell overexpressing ATPase dead version, and you can see it no longer makes stress granules, okay? So that fits with what I just told you, is that all it has to do is be able to bind RNA. It doesn't have to be able to hydrolyze ATP and act as a motor. But if it can't bind RNA, that's this cell here, you can see it still makes stress granules very similar to the cell that can make, that doesn't overexpress this. So what this suggests then is that all you need to do is to bind RNA, and you can block the formation of RNA granules. Now, all of this is done in cells, so we would like to see if we can extend this to a purified system. So in order to do this, what we did is we actually asked, can we demonstrate these same kinds of properties in a completely reconstituted system with recombinant EIF4A and purified RNA? And so the very simple experiment is to ask, okay, if we can make RNA self-assemblies in vitro, how does EIF4 affect those in the presence of ATP? And we were helped here with Jerry Peltier, who's a longtime biochemist who's done a lot of terrific work on EIF4A in his career. All right, so here's what that experiment looks like. Here what we're doing is I'm showing you the RNA itself is fluorescently tagged, so we're looking at the assembly of RNA molecules. And you can see that in the presence of just ATP and RNA, over time, we will form these RNA-based condensates. If we then add 4A in the presence of ATP, you can see that you greatly reduce the ability of those to form. So that recapitulates exactly what we see in cells and says this is a direct effect of EIF4A. If we add an analog of ATP, which can bind to 4A but not be hydrolyzed, you can see you still form RNA condensates, but they're reduced compared to the absence of 4A. So we can see that then, similar to in cells, you don't need to hydrolyze ATP to reduce RNA condensation, although the ability to hydrolyze ATP actually increases the efficiency, probably by allowing multiple turnover events. And then finally, if we add this drug hyperistanol, which blocks 4A from binding to RNA, you can see that we form RNA condensates very similar to the absence of 4A at all. So what this means then, and the way we think about this, is that EIF4A is preventing RNA condensation really is an ATP-dependent RNA binding protein. And that when you have RNAs which have exposed surfaces, those exposed surfaces can do one of two things. They can interact with other RNA molecules to promote the formation of RNP granules or stress granules in the case of the model system, but I would argue this occurs in many other granules as well. Or they can bind to EIF4A, which competes for those RNA-RNA interactions, destabilizes them, prevents RNA self-assembly, And then following ATP hydrolysis, EI4A is released, allowing the RNA to go about its business and allowing EI4A to recycle and carry out this role again. Now, one of the things that's very striking about this to us is how similar this is to HSP70. So the way we think about HSP70 and the maintenance of proteins and preventing protein aggregation is that when proteins are misfolded, they expose regions prone to promiscuous interactions, short hydrophobic stretches. Those hydrophobic stretches can then interact with each other to drive protein aggregation, or they can be bound by HSP70 in the presence of ATP to prevent aggregation, and then subsequently ATP hydrolysis leads to HSP70 release and possible protein refolding. And so in fact, what I'd like to suggest is that just like we're really used to thinking about protein chaperones, which limit inappropriate promiscuous interactions in cells to allow proper protein function and prevent protein aggregation, that we should expect that there's a whole family of RNA chaperones which function in the exact same way, and that probably the most prevalent and most important of these is going to be EIF4A simply because of its abundance in cells. This suggests three different implications. One is that cells have to have ways to prevent RNA condensation and preventing these RNA molecules from forming these kinds of promiscuous interactions. And I've talked about EIF4A, but there are certainly other dead box, quote, helicases involved in this as well. And we've been studying other molecules which play a role, things such as nucleases, which will reduce the RNA concentration to prevent condensation, or things like RNA modification enzymes like ADARs, which modify duplexes to destabilize them and can also limit the ability of RNAs to self-condense. The second implication, of course, is that evolution is going to take advantage of this fundamental biophysical property of RNA molecules to self-assemble to create specific interactions and specific RNP granules. Liz will talk more about this, I think, in her talk, to help impart biological functions. And I think a really nice example of this comes from Anna Fruzzi's lab, where she showed that base pairing between the Oscar 3' UTRs in trans help promote dimerization of Oscar messages and formation of an efficient RNP transport granule. Then I think that the implication that we've really tried to follow up on then is that, we're used to thinking about RNA as being, protein as being misfolded, but maybe we should also think about RNA just like misfolded proteins, that RNA can be prone to aggregation and can form RNA-based assemblies in cells, which in some cases may actually be toxic and really be contributing to some kinds of misfolding diseases. And, it's really striking to me that many of the proteins that we all study in protein misfolding diseases and form aggregates are very often RNA binding proteins. So TDP43 is one of the most prevalent proteins found in misfolded aggregates in cells, and it's a really terrific RNA binding protein. And in this light, my lab has then gotten interested in the protein tau. And we've begun to ask the question of whether tau aggregates are actually RNA protein granules. So, okay, so I just want to remind everybody what tau is. Tau is a protein that's expressed primarily in neurons in human cells, and it forms aggregates in a wide variety of neurodegenerative diseases. The most classic is in Alzheimer's disease, where the formation of A beta plaques can sometimes leads to a downstream formation of these tau aggregates in cells, which are involved in the toxic effects of that disease. But also diseases like chronic traumatic encephalopathy, where repeated blows to your head, if you're a football player, or can lead to formation of tau aggregates. This also happens in genetic conditions where specific mutations in tau lead to the formation of tau aggregates in a wide variety of specific diseases. So we got interested in trying to ask, what are these tau aggregates really? And I want to point out this is a collaboration between a very talented MD PhD student in my lab, Evan Lester, and Stan Prusner's lab at UCSF, primarily involving Felicia, Amanda, and George. So why do we even think about this, right? And so, people have been studying tau for decades. And one of the things that people have observed about tau is that in order to take a purified tau protein and make these tau fibers, you need to add some kind of polyanion. And most people do this experiment. They add 20% heparin to their test tubes. But to my knowledge, cells don't have 20% heparin in them. And then the other thing that they can add is RNA. And then RNA is really great at promoting the formation of tau fibers in vitro. And so maybe, in fact, then in cells, tau aggregates might be forming with RNA. And if you actually look in patient brains, there's this old paper from Ginsberg et al. That tau aggregates will stain positive for acridine orange, which is a nucleic acid dye, suggesting maybe these are actually containing RNA molecules. So we set out to try to see if there were RNAs in tau aggregates and what they were. We started out using a model system where tau forms these beta sheet fibers, which can propagate just like a prion. And if you take a brain extract from a mouse, which is making tau aggregates, and you transfect it into cells which express tau, it will trigger the formation of these tau aggregates, as you can see here, labeled in green. And I just want to point out that there are not only cytoplasmic tau aggregates, which are what is described primarily in the literature, but there also are nuclear tau aggregates, which you can see here within the nucleus. And these are not only seen in this cell system model, they're also seen in numerous mouse models and even in human biopsies in some cases. These aggregates contain RNA by a number of different criteria. The simplest one I'll show you here is if we lyse these cells and then we purify the tau aggregates and stain them for fluorescent dyes for RNA, you can see that this tau aggregate is really brightly lighting up with an RNA dye. So what RNAs are in these assemblies? So what Evan did is he worked out ways to purify them. In this case, he used a cell sorting method. And then once he sorted them, he sequenced them. And what we were surprised to find is that tau aggregates are really enriched for SNO RNAs and snRNAs. So just reminding you, SNO RNAs are typically found in the nucleolus and play a role in the biogenesis of ribosomal RNA. And snRNAs are typically found in the nucleus, particularly in what are called nuclear speckles, and play a role in pre-mRNA splicing. This enrichment of snRNAs and SNO RNAs in tau aggregates is not only true in these tissue culture models of tau, but we purified tau aggregates from mice that form tau assemblies. And you can see that, again, the snRNAs are the most highly enriched RNA in those tau assemblies. So the presence of all these snRNAs and SNO RNAs, these are normally nuclear RNAs. And so this made us start thinking about these nuclear assemblies of tau that we were seeing in our tissue culture model, and also we would see them in the mice. And snRNAs are typically found in what are called nuclear speckles, and SNO RNAs are typically found in the nucleolus. So we stained with markers for the speckles and for the nucleolus to see where these tau aggregates were. And what you can see is that they're primarily, essentially all, found in what are called nuclear speckles. So what I'm showing you here, this antibody SC35 is a marker for nuclear speckles. And you can see that in cells that have nuclear tau aggregates, like these dots here, you can see overlap really terrifically with nuclear speckles. So just to remind you, nuclear speckles are these transient membranous organelles that contain nascent transcripts and the splicing machinery. And they're thought to play roles in transcription of RNAs, splicing, and also the regulation of RNA export. These nuclear tau aggregates being in speckles is not unique to this cell line. If we look in mice, this is a neuron, a nucleus from a neuron showing tau aggregates within the neuron, and they're overlapping with these markers of nuclear speckles in this case, which is just oligo-DT RNA. So nuclear speckles, so tau aggregates end up in nuclear speckles. And we think they alter the function of those speckles in ways I'll talk about in a second. Right. So we also saw a surprising thing in these experiments. When we were looking at this antibody SC35, you can see here that in the wild type cell, it's all found in nuclear speckles in these assemblies in the nucleus. But if you had a cytoplasmic tau aggregate in this cell down here, the protein moves from the nucleus to the tau aggregate. And there's enough protein moving that it actually depletes the nucleus from the concentration of the protein in the nucleus. So we've since gone on, I don't have the slide to show you this, but we've looked at now probably 20 different proteins. And I can tell you that some, but not all, components of these nuclear speckles move into cytoplasmic tau aggregates. But many proteins, at least several proteins that are involved in pre-mRNA splicing, mislocalize to cytoplasmic tau aggregates, suggesting that there's a change in the concentration of key splicing factors in the nucleus, which might affect RNA processing. And so kind of the model that we started to come up with would be that, tau aggregates actually contain RNA, that RNA either directly or indirectly can titrate in RNA binding proteins. And that by doing so, will change that sequestration will change RNA processing in the nucleus. And I think before we went much further, we wanted to see if this was really true in human patients. And so what we did was we actually obtained brains through the brain bank at UCSF to ask if we saw similar things in biopsies of patients that have tauopathies. And so this is an example from an individual who had a cortical basal degeneration. This is a tauopathy driven by genetic mutations in tau. And what you can see here is that in the age-matched control, here we're staining for this SC35. You can see it's all in these little dots in the nucleus, which are speckles. In the patient, the green is these tau aggregates, which are forming in the cytoplasm. And you can see those tau aggregates, just like we saw in our cell line and in mice, which I haven't showed you, they now accumulate this SC35 staining as well. That's true not only in cortical basal degeneration patients, but it's also true in a number of different human tauopathies. These are 3 different patients who have Alzheimer's disease. You can see in age-matched controls, the SC35 staining is all in the nucleus. In these patients, it's depleted from the nucleus. You'll see nuclei with almost no protein in it. And it now accumulates in these fibers, which in many cases overlap with tau. This is also true in another tauopathy called frontal temporal lobe dementia in 3 different patients. So this is not something that's just seen in some weird cell line. It's actually seen in human patients. And so we really like to try to understand what's going on there. And this is something we're continuing to work on. One of the predictions of this model would be that not only are these proteins mislocalized to the cytoplasmic tau aggregates, but because they're mislocalized, they're going to actually change the splicing of RNA. And one of the things that's been seen in many, when people have studied Alzheimer's disease brains, is there are a tremendous number of splicing alterations. And so what we have been able to show is that mice that make tau aggregates or cell lines that make tau aggregates, that by itself is sufficient to change the splicing of a large number of different genes. It's very difficult to relate this back to what we see in human patients, because in human patient brains, many things have happened by the time you're actually getting a biopsy to sequence, including the patient has passed away. There's been a lot of neuron loss. But the important thing is that we can actually show that there are changes in splicing, which are driven solely by tau aggregates. All right. So I think I'm closing. I just want to make a few kind of comments. One of the key questions we're trying to answer now is, we see RNA in these tau aggregates, but is it really important for their formation? Or is it just a bystander? I think this is really a critical question for understanding how tau aggregates form in disease. My bias is that, in fact, these are very much like RNPs that we've studied in other contexts, that not only is the RNA there, but it's actually part of the assembly and plays a role in actually forming them. So the other thing I want to point out is that we're suggesting a converging mechanism of neurodegenerative disease. We're used to people who've been studying ALS have shown that TDP-43 accumulates in these cytoplasmic RNP granules in disease. And when it's titrated from the nucleus, it changes RNA processing. And so what I would suggest that that's also occurring in tauopathies, where tau aggregates actually are sequestering a number of RNA binding proteins. And that triggers changes in RNA processing, which actually contribute to disease. As I mentioned, one of the key questions is RNA really just recruited to fibers that form, or is it actually part of the formation of assembly? And one of the reasons that I really like the idea that the RNA molecules play a role in this assembly is twofold. One is the role of RNA in vitro in promoting fiber formation. But I just want to show you some images. If you form fibers in the presence of RNA, and that RNA is fluorescently tagged, these would be tau fibers here shown in green. This is an RNA that we added to drive their formation in red, and you can see that they co-localize all along the fiber. And that's also true if you look in our cells. So this is actually one of the RNAs that we see enriched in tau aggregates. And here we're zooming in on a particular tau fiber, and you can see the tau fiber and the RNA are really coincidence all along its length. It's not that it's just a nucleating event and then gone. So a key question we're trying to answer then is, does this RNA actually play a really important role in the formation of these fibers? So I'm just going to end there with, I think two important punchlines for you to take away. One is that RNA is really prone to self-assembly and that cells modulate that through a number of different mechanisms analogous to protein chaperones, but for RNA. And EIF4A is probably the big dog in the room because it's very similar to HSP70 and how it works. And then the implication of that is that RNA aggregation might be similar to protein aggregation in some contexts. And we think at least one place where that's looking to be true is tau aggregates, where tau aggregates are not just protein assemblies, they're actually tau RNA protein assemblies. And that may play a role in not only their formation, but also how they drive toxicity in human neurons. Okay, so I'm going to stop there. I'm sorry I went a little bit over. And I'll stop sharing my slide.
Okay, thank you. Thank you, Roy, for the very nice presentation. Let's move on to Elizabeth. Dr. Elizabeth, could you share your screen, please? And again, thanks for accepting the invitation. Let's keep the questions for the end of the presentation.
Sure. Okay, let me go ahead and...
Excuse me, let me just give a brief introduction about Dr. Gavis. She got a major at Yale in biology, and then she got the MD and the PhD at Stanford, and then the postdoc was in the Roots Lehman Lab and the MIT, and then she moved to New Jersey, right? To Princeton University, and she's a professor since 1994, and she's also editor of several journals, including the G3. It's a really important journal, and right now I'm not going to talk about your research interest because you represent it right now. Thanks again.
Let's see. Right. And be able to see my play thing. Okay. Can everybody hear me okay? All right. So thank you very much, Fernando, for this really lovely invitation. It's really a pleasure to be able to talk with you guys. I wish I could be there in person. Obviously, that's not possible. So we'll have to do the best we can virtually. So the work in my lab focuses on post-transcriptional mechanisms that exert exquisite spatial and temporal control over gene expression during development. And post-transcriptional regulation is especially important at the very earliest stages of development. And that's because in all animals, or nearly all animals, there's a delay between the time of fertilization and the onset of transcription of the embryo's own genome. And for many animals, including many of the model organisms that we study, and in particular the fruit fly Drosophila, which is my favorite model organism, there can be a delay of many cell divisions before the embryo's own genome can be transcribed. And so the very early stages of development of these animals rely entirely on stores of mRNAs, we call these maternal RNAs, that are produced during oogenesis, and they're stockpiled in the egg so that they're ready for the embryo to use at the time that the embryo is fertilized and can run the early developmental programs of these embryos. Now, because these RNAs are present in the embryo from the get-go, from the very beginning, any use of these RNAs must be controlled post-transcriptionally. So for example, the where and when proteins are translated from these RNAs has to all be controlled at the post-transcriptional level. So this is sort of a classic example from Drosophila of two RNAs that are post-transcriptionally regulated at the level of localization and translation. These are early Drosophila embryos, newly fertilized. This is an RNA encoded by the bicoid gene. This is an RNA encoded by the nanos gene. These RNAs are localized at opposite ends of the embryo. They're physically localized in these places, and this happened way before the embryo was actually fertilized. And then these RNAs are translated in these local positions to produce proteins that diffuse to make these lovely gradients that are involved in patterning of the anterior to posterior head to tail body axis of these embryos. So these are RNAs that are post-transcriptionally regulated by their localization and their local translation. Now, this kind of regulation certainly isn't specific to maternal transcripts, and it's not limited to early embryos, but plays an important role in particularly polarized cells, such as neurons. And this is a hippocampal neuron in culture. And Roy already mentioned that RNAs, these are little RNA particles that are trafficked out into the dendrites where they are held. The RNAs are held until synaptic stimulation occurs. These RNAs can then be translated on site. And this type of local regulation, of this localization and translational control is very important for synaptic plasticity associated with learning and memory. And my lab is actually interested in neuronal RNA trafficking. We study it in the context of dendrite morphogenesis, and I'm not going to talk about that. But what I'm going to focus on today are RNAs, in addition to nanos, that are localized in this special region of the posterior pole of the Drosophila embryo. And so I'm going to focus on RNAs that are enriched in a special cytoplasm that's called the germplasm that resides at the posterior pole of the embryo. And the germplasm specifies the formation and the fate of the germ cells, which are going to bud from the posterior of the embryo. And I'll explain that a little bit better. But first, I just want to kind of catch everybody up on the sort of early divisions of the Drosophila embryo because they're not terribly typical. So in Drosophila, the embryonic nuclei divide, but their divisions are not followed by cell division. So they're not followed by cytokinesis. And these nuclei will divide, and after the 8th division, the nuclei will migrate to the periphery. And those nuclei that encounter this special cytoplasm called the germplasm will be induced to push out against the plasma membrane. And then they will ultimately pinch off to form these cells that I'm going to call pole cells because they're at the pole, but these are the germ cell progenitors. These are the cells that will give rise to the eggs or the sperm of this animal, depending on whether it's male or female. And these cells are born outside the embryo, but during gastrulation, they will migrate inside to coalesce with somatic cells to form the gonad. And it's only after these pole cells form that the remaining nuclei will become cellularized to produce a blastoderm embryo. Now, the germplasm, we now know, harbors several hundred maternal transcripts. These transcripts become very highly enriched in the germplasm, and many of these transcripts are packaged or become partitioned into non-membrane bound condensates that we call germ granules. And this is in sometimes you'll hear the word polar granule referred to the Drosophila germ granules, but more generally, they're called germ granules. And these germ granules deliver, so sorry, I'm just showing the images. So these are images of these germ granules, and they provide a mechanism to deliver these maternal RNAs to the pole cells, because like the embryo, these pole cells are also transcriptionally quiescent for the beginning of their life. And so they need their own maternal supply in order to run their early developmental program. And these germ granules provide RNAs to the germ cells. Now, I should also mention that the germ plasm is, in ways that we don't understand, it is both necessary and sufficient to induce the formation of these germ cells. If you remove it from the embryo, germ cells won't form. This will not happen. If you transplant the germ plasm to the anterior of the embryo, germ cells will form at the anterior of the embryo, and you can actually transplant those cells into a host embryo, and they will develop into perfectly functional germ cells. So our lab has really focused on 4 transcripts that are components of the germ plasm. They're highly enriched in the germ plasm, and they're components of these germ granules. And they are nanos, polar granule component, which I'll refer to as PGC, germ cellus, which I'll call GCL, and cyclin B. And I'm mentioning these right now because I'm going to refer to these transcripts and multiple times during my talk. And I just want you to realize what I'm talking about here. So these are all RNAs that play a role in subsequent germline development. They are partitioned into these non-membrane bound organelles or germ granules, and they are delivered to the germ cells. Now, it's hard to figure out who was the first person to actually discover the germ granules, but there's I think what I've been able to find is a paper from, which I can't read because it's in a different language, by Ritter, who identified RNA staining granules in the germ cells of the midge Chironomus. But I think the first real connection between these germ granules and germ cells and the germ plasm was made by Hegner in this monograph. And he writes that at the posterior of the eggs of Calligrapha multipunctata and allied species, there is a disc-shaped mass of granules which stain like chromatin, and that's because they have RNA in them. I have called this the pole disc. When the cleavage nuclei reach the periphery of the egg, they fuse with the superficial layer of cytoplasm everywhere except at the posterior end. Cell walls, when I think he means cell membranes, then appear and a blastoderm is formed. When the cleavage nuclei, which reach the posterior end of the egg, encounter the pole disc granules, they gather these granules about them and continue their migration. Cell walls or cell membranes are formed, and they finally come to lie outside of the egg. And essentially, this is what I just showed you in my previous slide, right? So he also did some experiments where he pricked the eggs of this beetle, and he let the germ plasm flow out and the germ granules flow out, and he showed that he did not, these animals no longer form germ cells. So he's really the person, I think, who made this first connection, and people have been studying these granules ever since. So germ granules, though, are not specific to insects. In fact, they're hallmarks of germ cells throughout the animal kingdom. They are quite well known for their, the germ granules in C. elegans have been quite well studied. These have been a really nice model for studying liquid-liquid phase transitions and formation of RNA condensates. These are called P granules, and they ultimately become segregated to the cell that will give rise to the germline in C. elegans. Germ granules are found localized in xenopus eggs, and these are in zebrafish, and you can see they become inherited by cells here again that will give rise to the germline. Now, in mammals, such as the mouse, germ cells are not specified through the inheritance of a maternally deposited germ plasm. Germ cells are instead specified through inductive mechanisms, through signaling mechanisms between extraembryonic and embryonic tissues. But once those germ cells have been specified, germ granules will begin to form de novo in these already specified germ cells. And there are a number of shared proteins and RNAs among the germ granules of all of these different animals, suggesting that there's a commonality of function. So I just want to make it clear that what I'm telling you is not necessarily specific to Drosophila, but I think lessons can be learned for germ granules throughout the animal kingdom. So before I go any further, I just want to thank the people whose work I'm going to be talking about. The story will start with work that was done by Sean Little, who was a former postdoc working both with myself and Eric Wieschaus, my colleague here at Princeton, together with Christina Sinsheimer, a postdoc in my lab, and Jack Lee, a former graduate student. And Sean now has his own lab at University of Pennsylvania. And this work was then followed up by postdoc Matt Napelko, who's done the super resolution studies I'm going to show you. And he has now his own lab at Kean University. I'll also talk about work from former graduate student Whitby Eagle, Catherine Eichler, and current student Catherine Doherty. So this is the team. Now, although I've told you that the germplasm functions in the embryo, it's assembled much earlier in time during oogenesis, so in the egg while it's being produced in the mother. In Drosophila, the oocyte doesn't develop on its own, but it develops in a cyst connected to 15 sister cells. So this is the oocyte. These are the sister cells called nurse cells. All of these cells derive from the division of a single precursor with incomplete cytokinesis. So they stay connected to each other by cytoplasmic bridges that allow the passage of molecules like RNAs and proteins and mitochondria. The maternal RNA, so that the oocyte becomes meiotic and enters meiosis, and these nurse cells, by contrast, become polyploid, highly polyploid. And this allows them to serve as synthetic factories that are going to produce the maternal RNAs that are needed for the growth and development of the oocyte and ultimately the early stages of embryogenesis. And the key player in the initiation of the germplasm is an RNA called OSCAR, which Roy already mentioned. And OSCAR is, like all the other maternal transcripts, is made in the nurse cells and is packaged into ribonucleoprotein particles. And these particles are then transported into the oocyte. They become associated with kinesin motors and can travel on oocyte microtubules towards the posterior of the oocyte, where they accumulate in a mechanism that we don't really understand. They form actually quite large granules at the posterior. And OSCAR protein then, once the RNA arrives at the posterior, OSCAR is translated, and it's only translated at the posterior. So it's really the localization of OSCAR and the translation of OSCAR at the posterior that sets up the position of the germ granules. So OSCAR protein is produced, and OSCAR can recruit additional proteins like the helicase vasa, which is one of the DDX proteins in mammals, tutor, the peewee protein aubergine, and others. And work from Ruth Lehman's lab has shown that OSCAR can induce phase transition. And so the idea is that OSCAR is promoting condensation of these proteins to form protein granules. So protein components of these germ granules that are not, as I said, condensing at the posterior. And these are going to essentially form scaffolds into which the RNAs that will populate the germ granules are going to accumulate. Now that's going to happen subsequently. So what I'm going to show you now in a movie is that the nurse cells will initiate a cell death pathway, and they will contract and extrude their contents into the oocyte. The oocyte microtubules will become reorganized in bundles to mediate streaming of the oocyte cytoplasm that's going to mix the incoming nurse cell contents and the oocyte with the oocyte contents. And the oocyte will grow very large, and it's during that time that the germ granule RNAs will be entering the oocyte and becoming incorporated into the germ granules. So this is just a movie to show you the process. These are the nurse cells here. This is the oocyte, and what you're going to be looking at is autofluorescence of the yolk. So the nurse cells you can't really see the contraction, but they're contracting, they're dumping their contents into the oocyte. The oocyte microtubules are mediating the streaming, which is essentially mixing up all the nurse cell contents with the oocyte contents. And the oocyte grows very large at this time, and the nurse cells are dying off. And it's during this period that RNAs are going to be accumulating in those germ granule scaffolds that are pre-assembled and pre-localized at the posterior. So maternal, as I said, all essentially the maternal RNAs are getting dumped into the oocyte, And there's this special subset of them that are going to become enriched at the posterior of the oocyte. So, sorry, my little, I'm having trouble because I can't see the edge of my slide because of the pictures of the, so I can get rid of this. There we go. Okay. So these RNAs will enter the oocyte and they diffuse, they essentially become distributed throughout the oocyte cytoplasm and will start to accumulate. We can visualize them using live imaging methods. We can visualize them accumulating in the germ plasm in association with these pre-localized germ granule protein scaffolds. So I should also mention that this process is actually fairly inefficient. So although the RNAs become highly concentrated at the posterior, it's only a subset of all of the total RNA that becomes localized. So for example, for the nanos RNA, only about 4% of it actually ends up in these germ granules, and very similar, we get very similar values when we look at some of the other RNAs like PGC, cyclin B, or GCL. So they become very highly enriched here, but it actually represents a very small proportion of all the total of each of these RNAs. And the remaining RNAs that are unlocalized ultimately become translationally repressed and will become degraded over time. The other thing that I think I neglected to mention at the beginning, which is important, is that there are several hundred RNAs that we believe are actually becoming localized in the germ granules during this time. So to better understand what was going on, what do I mean by this localization process, this entrapment process, how are the RNAs becoming associated with the germ granules? Sean decided to, together with Christina and Jack, decided to take a quantitative imaging approach to actually sort of really look in detail at what was in these germ granules. And so in this experiment, this is a single molecule fluorescence in situ hybridization experiment in which he's detecting nanos with, and PGC with sets of small oligonucleotide probes that are individually labeled. And there's a large number of probes decorating each RNA. So this gives us very high signal to noise and high resolution. And what Sean was able to show was that these RNAs travel within, they enter the oocyte and they travel within the bulk cytoplasm as single RNA particles. So you may be able to see these individual little magenta and green dots. Each of these represents a single RNA molecule, likely packaged with proteins to form a small ribonuclear protein particle, but each containing one RNA molecule. And you can see that they never overlap. These are two different RNAs, never overlap within the cytoplasm. But at the posterior, you can see very nice co-localization in these very bright foci that are the germ granules. And what we can interrogate individual granules and ask how many of different RNAs are in those granules. And this is just one example where Sean has interrogated granules and asked how many PGC molecules are there and how many nanos molecules are there in any one granule. So he detects there are granules that contain varying numbers of PGCs, but no nanoses, varying numbers of nanoses, but no PGCs. And then there are many granules that contain varying numbers of PGC and nanos RNAs. And we can do this with different pairwise combinations of RNAs, like we can do similar experiments with these and GCL or cyclin B, and we get very similar results. And so this suggests to us, or indicates to us, that these granules are quite heterogeneous, both in the different RNAs that they contain, as well as the number of each RNA that they contain. Now, one of the crucial things is that these granules, these RNAs, do not accumulate like this. We never see their co-localization unless we have those pre-made germ granule protein scaffolds. So in mutants that don't make, that are mutants for the Oscar protein or VASA or any of those other proteins that are part of these granules, the RNAs do not accumulate this. We never see co-localization. We never see these high levels of accumulation like at the posterior. We can also interrogate each granule and ask, we can mark the granules with, for example, GFP tag proteins for the different protein components of the granule, and we can interrogate the RNAs together with those proteins. So this is an experiment where we're looking at VASA, one of the protein components, together with nanos. And there are granules that contain VASA but no nanos, and presumably they contain other RNAs. But every time we see nanos in one of these bright foci at the posterior, it's always associated with a VASA. So the granule proteins are absolutely obligate for these RNAs to accumulate at the posterior. Now, Matt Napelko decided to look in more detail by using super resolution imaging to see how these RNAs were actually arranged within the granules. And so this was the confocal image I showed you before, but this is SM Fish experiment where he's detecting nanos and PGC together with the Oscar protein that's tagged with GFP, and that's going to be blue. And these are SIM, structural illumination microscopy images from the middle region of the oocyte. And you can see again these individual single transcript particles, completely non-overlapping. At the posterior now, you start to see significant overlap between these two RNAs as well as together with the Oscar GFP marker. And if we zoom in on one of these granules, this is what we can see. The granule again marked in blue with Oscar. And what we've observed was that the RNAs are not intermingled within the granule, but yet they sort out from each other. This is actually a group of 4 nanos RNAs. This is a group of 3 RNAs. And so we call these homotypic clusters. And one way you can think about this is that the RNAs are essentially, living in the same house, but they're occupying separate bedrooms. So they're forming these distinct homotypic clusters. So Matt did a very intensive study to try to understand how these clusters were forming and how these granules were assembled. And I'm just going to summarize all of his work together with some work from Sean on that, and it's all in the data are all in these papers if you want to take a look, but I'm just going to summarize all this on this one slide. And so the idea is that I've already told you that these RNAs are circulating within the cytoplasm as single transcript RNPs. And at some frequency, they make encounters with these pre-localized germ granule protein scaffolds. And these are random encounters. And at some frequency, they may become incorporated into a granule where the RNAs, these single transcript RNPs will serve as seeds to recruit additional transcripts, like transcripts to form these homotypic clusters. And we think that this is a stochastic process, and that this stochasticity of how frequently and which RNAs populate different granules leads to this type of heterogeneity that we observe. Now, the granules grow over time through the accumulation of additional granule proteins. Those proteins are continuing to be made organized at the posterior, and they will essentially embellish the granules. The granules will grow. And this growth of the granule protein scaffold is required for the continual growth of these homotypic clusters. New granules will also continue to form, and they will continue to be seeded. Now, what Matt noticed was that over time, the frequency with which the different RNAs populate the granules doesn't change very much, but what does change is the amount of RNA per granule. So the clusters are growing, and it's really, we think it's this sort of self-recruitment and this growth of the homotypic clusters that's really what drives the accumulation of the RNAs at the posterior. And we can change the amount of RNA for example, we can change the amount of nanos RNA in the oocyte by playing genetic tricks. And it doesn't really affect the frequency with which nanos populates the granules, but what it does is it affects the amount of nanos RNA in the clusters. So again, it really suggests to us that it's this self-recruitment and this clustering function that's really driving the accumulation. But sometimes we do see a granule that might have 2 clusters of the same RNA. That's not frequent, but we do see it. But if we sort of quantify the average amount of RNA per granule and take the ratio of that to the ratio of clusters per granule, it's always more than 1. And that tells us that the RNAs are more likely to join an existing cluster than to seed a new one. So if an RNA enters a granule and there's already a cluster, it's much more likely to join an existing cluster of itself than to start a new cluster. So this model predicts that there are features of the RNA that are involved in targeting the RNAs to the granules and allowing their incorporation into the granules, and I'll call those targeting elements, as well as features that mediate the clustering or the self-recognition and the clustering of these RNAs. And I'll call those clustering elements. And the obvious place to look was in the 3' untranslated regions of these RNAs. So I had shown many, many years ago that the 3' UTR of nanos, for example, was sufficient to target a reporter RNA to the posterior of the oocyte and in the embryo as well. And what Whitby did in this experiment was to actually ask whether or not those 3' UTRs are sufficient to get the RNAs into the granules. And so this is a GFP reporter that he's detecting using single molecule FISH probes, and he's appended different 3' UTRs to the GFP reporter, and then analyzed the localization of these reporter transcripts in transgenic animals. So he's this is the GFP reporter with the nanos 3' UTR. This is the one with the PGC 3' UTR, and we're using, in this case, the native endogenous nanos RNA just as a way to visualize the RNA granules, the germ granules. So you can see that the nanos 3' UTR, this RNA with the GFP RNA colocalizes very nicely with nanos in these white foci at the posterior. These are the germ granules. And so he can show that these 3' UTRs are sufficient to get the RNAs into the granules, but they don't mediate the self-recognition that is needed for clustering. So this is an experiment that's going to show that, and this is just a control that I need to show you at the beginning. This is just a control to verify that our imaging conditions allow us to detect true colocalization. So this is detecting nanos RNA with probes of alternating fluorophores, and we should see just a single, in a super resolution image, we should see just white, right? The merge of the two colors. And if our imaging conditions are good, and we do, we see these are we are detecting the nanos RNA in these granules, and we're seeing essentially white. And I'll show you why this was an important control. So this is an experiment in which Whitby is visualizing the GFP reporter with the nanos 3' UTR together with the native nanos RNA, and using probes that detect only the coding regions. So there's no crosstalk between these. So we're detecting the nanos coding region and the GFP coding region. And what you can see is that although this nanos 3' UTR can get this reporter into the same granule as the native nanos RNA, they sort out from each other. This RNA is forming its own clusters. This RNA is forming its own clusters. So they're not forming mixed clusters. And we see the same thing with PGC. Again, the 3' UTR can get it into the same granule, but the RNAs do not, they sort out from each other. These RNAs do not recognize each other. And Tatjana Tresek in Ruth Lehman's lab has done other experiments where she's inserted various tags into the nanos RNA and shown that the RNAs with these different insertions they won't recognize the endogenous nanos RNA. We've also shown that the 5' UTR is not mediating this recognition. And we've done this for additional RNAs as well. So it seems that the 3' UTR is nor the 5' UTR nor the coding region per se contain specific features that mediate self-recognition, and rather it's some sort of holistic property of the RNA itself that's mediating this self-recognition. Now, the 3' UTRs don't mediate self-recognition, but we do think they play a role in clustering. And that comes from some experiments that both Matt and Whitby did looking at deletions of the 3' UTRs of nanos and PGC that affect their accumulation in the granules. So I had made deletions of nanos years ago to try to understand better if I could define parts of the nanos RNA that were involved in localization. And Matt sort of revisited these constructs, which I had saved for probably 30 years or something. Unbelievable. I still can't believe I still had the flies, but we did. And he found that this one particular deletion was especially interesting. So this experiment is essentially looking at flies that express either wild type nanos RNA or nanos RNA that contains this deletion of about 90 nucleotides in its 3' UTR. And we assay the ability of the RNA to co-localize with a granule marker, like Oscar, for example. And we set that to 100% for wild type. For this nanos that has a deletion in it, it co-localizes with the granule markers just fine. It gets into granules at the same frequency as the wild type, but the amount of RNA in each cluster goes down. It's close to like 45% reduction in the amount of RNA per cluster just by deleting this 90 nucleotide region. And you can see the difference in this intensity histogram of the amount of RNA per granule. This piece alone does nothing, and it's presumably because it's lacking the sequences that target to the granules. And Whitby found something similar with the PGC3' UTR. These experiments were done a little differently because he is using a GFP reporter to assay localization, but the wild type 3' UTR, again, we set the ability, we set this to 100%. This is a deletion of 59 nucleotides in the PGC3' UTR that has very little effect on the ability of the RNA to get into the granules, but it reduces the amount of RNA in the clusters by 50%. And again, so like with nanos, this isn't the whole story because it's only a 50% reduction. And so presumably there are other elements in other parts of the 3' UTR. This element on its own does nothing. In 3 copies, it does nothing. But if we append extra copies to this part of the RNA, we can actually restore near wild type cluster size. So Matt wanted to know whether these elements are specific for the RNAs or whether they could be exchangeable. And so he did a couple of chimeric swap experiments making chimeras. And so this experiment, he has replaced the nanos clustering element with this clustering element from PGC. And the assay here is he's just measuring both the maximum and the average cluster size that is produced when you express this wild type version of nanos, which is here. This is the version with the deletion, which is way down here. It drops the average and the maximum size of the clusters that can form significantly. But if you put in this, you replace this part of nanos with this PGC element, it bumps it up, right? So you can restore at least partially the sizes of the clusters by substituting this PGC clustering element. And he did a similar experiment with PGC in which he replaced the PGC clustering element with the nanos clustering element. This is the behavior of the wild type RNA. This is the deletion. And this is the RNA, chimeric RNA that has both the nanos clustering element, and it really pushes it up closer to the level of nanos. So just want to sort of summarize some of the things that I've just told you and also some data that I haven't really had time to tell you about. I've told you that the 3' UTRs seem to have germ granule targeting and some type of clustering function, but they don't confer self-recognition, nor does the 5' UTR, nor does the coding region. So that self-recognition is some sort of a holistic property of the RNA that we still have to figure out. The 3' UTRs, I haven't really shown you this, but I've sort of implied that the 3' UTRs contain multiple targeting and clustering elements, which we have shown, and that the clustering elements seem to act additively to tune the size of the homotypic clusters. So this leaves us with many questions that we still want to answer. We don't know how the clustering elements work. We'd really like to understand that. We would really like to understand what mediates self-recognition. And one other interesting question is that the RNAs, there are many germ granule RNAs that are circulating within the bulk cytoplasm. They never self-associate. They never associate with each other. They never associate, they never form clusters outside of the granule. So why do they only associate within the granule? What's special about that granule environment? So the ability to manipulate the size of the clusters, though, allowed us to ask whether cluster size actually matters. And so I'm going to show you that in this experiment that was done by Caroline Doherty. And she's asking now, what happens to germ granules when they contain nanos RNA germ cells that contain nanos RNA that have a reduced cluster size? So remember, nanos is, is going to be as a member of the germ, as a component of the germ granules, will be incorporated into the pole cells that form. Ultimately, these will give rise. These cells will ultimately have to migrate internally and coalesce with somatic cells to form the gonad. And I showed you way back at the beginning of the talk that the nanos RNA is translated at the posterior to form that protein gradient that's involved in patterning of the animal, but it has a second role in the germ cells themselves, which is why it needs to be incorporated and transferred into these germ cells. And so what Nanos does in the germ cells is it is in ways that we still don't entirely understand, it's involved in maintaining the transcriptional and mitotic quiescence of these cells and to promote their migration to the gonad. And in Nanos mutants, this mitotic and transcriptional quiescence of these cells is not maintained. The cells often initiate apoptosis, and they also have problems migrating to the gonad. So Caroline introduced, generated animals that express either a copy of the wild type Nanos RNA or where the only Nanos is Nanos that contains this deletion. And she analyzed what happened to the germ cells in their migration to the gonad. So this is a wild type embryo, and these are germ cells that are stained with a marker that shows up in this pink. And they are sort of lining up on the mesoderm here. They're going to coalesce very soon into the gonad. This is an example of an embryo that's where the Nanos RNA has a reduced clustering function. And these are germ cells that are lost. So it's a little hard to appreciate from this image. These are germs that are actually in a totally different plane from these germ cells that are migrating on the mesoderm, and it will coalesce to the gonad. So these are completely lost, and we can quantify this defect. So this suggests to us that homotypic clustering ensures that germ granules receive the requisite quantities of mRNAs that are needed for germline development. We also think they have likely other regulatory functions. What I haven't told you is that the RNAs that are in the germ granules are translated at different times. So Nanos is translated pretty much as soon as we can see it become associated with germ granules, we start to see Nanos protein. But for other RNAs like PGC and GCL, translation is delayed for many, many hours. And so, and they are also translated at different times. And so it's possible that the clustering may have something to do with this timing of translational control. And that's something that we're also interested in exploring. So just in the very last part of my talk, I want to, I've told you about the germ granules and their sort of assembly at the posterior of the embryo, of the oocyte, and their use in the embryo, but they are not the only granules in this germplasm. So remember, Oscar RNA is also localized in the germplasm, and it's actually the thing that sets up the assembly of the germplasm. It's localized at the posterior, but Oscar is not RNA. It does not go into the germ granules. Oscar protein assembles the germ granules, but Oscar RNA is in a separate granule. And this is just a single molecule fish experiment where you can really see that Nanos and Oscar RNAs do not overlap at all at the posterior of the oocyte. Oscar forms a granule together with a double-stranded RNA binding protein called Stauffen. Stauffen is important for its transport to the posterior and also remains associated with the RNA at the posterior. And the Oscar granules get very, very large. They can accumulate hundreds of Oscar transcripts. And this presumably facilitates the production of large amounts of Oscar protein to create the protein scaffolds. So I hate giving new names to things, but we called these granules founder granules just because we needed a way to distinguish them from the germ granules. So I'll refer to them as founder granules from now on. So these are the granules that contain Oscar RNA as opposed to germ granules, which are going to contain RNAs like Nanos and PGC. So we wondered whether the segregation of the RNAs into different granules is important, and we were able to drive Oscar into germ granules by removing Oscar elements that mediate its localization and replacing them with the Nanos 3' untranslated region, which will drive, as I showed you, the RNA into germ granules. So we can make germ cells now that have lots of Oscar RNA in them, and it turns out that this is really deleterious. So in fact, fewer germ cells actually form. These are germ cells at the posterior of that are newly formed at the posterior of the embryo. And there are many fewer in these embryos where Oscar is driven into the germ granules. And these germ cells behave very badly. So this is an embryo at the end of embryogenesis. These are the gonads, and it's a little bit hard to see, but they are populated by these very nice round germ cells. And the gonads of these embryos where Oscar was populating the germ granules are really depleted of germ cells, and the germ cells look really squirrely. So there, it's really, having Oscar in the germ granules is really detrimental. And we've shown that it's the RNA, not the protein, that's the toxic thing. And we don't know why, but it seems that it's really important to prevent Oscar RNA from becoming incorporated into the germ cells. So Kathy Eichler, grad student in the lab, noticed that Oscar RNA was getting degraded around the time of germ cell formation. And this is in contrast to the germ granule RNAs. So like nanos, for example, you can see these RNAs are persisting in the germ granules all the way through this process of whole cell formation. In contrast, Oscar is actually being degraded. And we think that it's this degradation of Oscar that's essential to prevent its accumulation in the whole cells, and then thus its toxicity. So during this period, about 60% of the maternal transcriptome is being turned over. And that's to allow for a transition to the zygotic genome. So there's a period during this time called the maternal to zygotic transition, where, as I said, the maternal transcripts become degraded. And I've drawn this as a single line, but it's really, there's multiple degradation pathways that kind of act at various times throughout this period. And then zygotic transcription is initiated. And the idea, again, as I said, is to turn over the maternal genome and replace it with the zygotic genome. And we thought that perhaps the degradation of Oscar was just part and parcel of this broader degradation process. And during this maternal to zygotic transition, remember I mentioned that there are a lot of RNA, like for all of the RNAs that are in the germ granules, it's only a very small percentage of the total RNA that gets into the germ granules. So for example, I mentioned that only about 4% of the total pool of nanos RNA is in the germ granules. The rest of it is throughout the bulk cytoplasm. All that RNA is getting trashed. It gets degraded, but the germ granule RNAs get protected. So we wondered though if Oscar was just being degraded as part of this general maternal zygotic transition. And so we did Kathy did a time course where she looked more carefully at the RNA over the early course of embryogenesis. And you don't have to worry about the stages here, but this is the RNA just after in a newly fertilized embryo. This is Oscar in the germplasm. This is in the founder granules. And this is the Oscar that is in the bulk cytoplasm that's not in the founder granules. And you can see that this RNA in the founder granules gets degraded well before the Oscar RNA in the bulk cytoplasm that's getting degraded as part of the maternal to zygotic transition. And we can quantify that over here. And so just to sort of summarize this or to put it in perspective, Oscar is getting degraded way ahead of the general degradation of RNAs that's occurring during the maternal to zygotic transition. So there's something special that's happening here. Now in eukaryotic cells, RNA degradation is generally mediated through the removal of the poly A tail by various nucleases, removal of the 5' methyl cap by the decapping complex that includes this protein DCP1, and then degradation by the XRN1 nuclease, which in Drosophila is called Pac-Man. So Kathy visualized Oscar RNA and the founder granules together with components of the degradation machinery, in this case DCP1. This is embryo almost just after fertilization, and you can see that there is really no overlap in the DCP1 and Oscar. So these founder granules do not co-localize with DCP1, but at the time that the nuclei start to move out to the periphery, we suddenly see the accumulation of DCP1 on the founder granules. And if we drive Oscar into germ granules, we don't see that accumulation of DCP1 with Oscar. So it's not Oscar per se, it's the granules, right? So the germ granules are not accumulating DCP1, but the founder granules specifically are at this time. And we looked at other components of the degradation machinery, and we see very similar behavior. And in fact, we see the accumulation of DCP1 with the Oscar granules, the founder granules, before we see accumulation of Pac-Man, which makes sense. And this is all in advance of the time where we start to see Oscar getting degraded. So, and the germ granules are protected Now, don't have time to go into another story, but the germ granules ultimately, much later in the pole cells will undergo their own maternal to zygotic transition, and germ granules will start to accumulate DCP1 in the degradation machinery, but not till much later. So there's this timing of association of the degradation machinery with these different granules that is somehow regulated, and in a way that we don't yet understand, but it's something, again, that we're quite interested in. So just to sort of summarize, I've told you about two different types of RNA granules that are populate this very dense cytoplasm called the germplasm in the Drosophila embryo. And there are the germ granules, which coordinate the delivery of many RNAs to the germ cells and protect these RNAs from degradation during the maternal zygotic transition in the bulk of the embryo. And then there are the founder granules, which localize OSCAR, allow for the formation of the protein components of the germ granules, but ultimately need to be essentially removed so that they don't, that the OSCAR, they need to essentially mediate the degradation of the OSCAR RNA so that it doesn't end up in the germ granules and cause toxicity. So I think this is really an example. I mean, you've heard about other types of granules from Roy today, and I think this is an example of, you know, how RNAs are compartmentalized within the cytoplasm according to their regulatory needs. And in the case of the germ granule RNAs, it provides a way to actually coordinate the regulation of those RNAs so that they can all get delivered, protected, and then delivered to the germ cells. So I will stop there, and thank you for your attention. I should also just thank the lab in general. I mentioned all the people who did the work, but I should also mention our funding through the National Institutes of Health. And thank you.
So thank you very much, Liz, for a very nice talk. So now, Tatiana, you control the questions. People, if you want, if someone would want to ask questions, please raise their hand or put the question in the chat, please.
Okay, everyone. So I had an early question for Dr. Roy from Luciana at the Virology Institute. So what is the role, if there's any, of RNA modifications like methyladenosine in granule formation?
So we don't really know very much about this. I think so probably the most data comes from studying M6A. So M6A, when it's on RNAs, we anticipated having two effects. One is it will recruit certain M6A binding proteins like YTHDF proteins, and that those can have a very small effect on targeting an RNA into a granule. For example, if we tether 25 copies of one of the YTH proteins on a message by artificial means, we can get a small increase in its presence in granules. So M6A can do that. If you get rid of all M6A, we've used embryo stem cells that are missing M6A modification enzymes, we don't actually see changes in RNAs getting into granules. This is in contrast to some results Sami Jaffrey has presented where he argues that he does see an effect. So I think, to me, the answer is probably that M6A makes a very small effect, but probably through the binding of these proteins. You know, I think there's an interesting question here, which is how do modifications of RNA sequences which change RNA-RNA interactions potentially affect these kinds of interactions, right? Which I think is maybe what the question is getting at. M6A should reduce base pairing a little bit, so it could weaken RNA interactions. We have not directly tested this, whether M6A modified RNA would be less effective at RNA condensation in vitro, but that's a prediction from its anticipated chemistry. It's really expensive to buy the relevant oligos to do that experiment, so we've never done it.
So anything, Fernando?
So I have a question, but I will wait for the others.
I think you can go ahead so people take courage.
Okay, so it's a kind of provocative question, because if I understand correctly, Parker. You said that at least for stress granules, it seems, my impression was, it seems that there is a natural tendency of the lot of mRNA to aggregate or assembly, and there is a kind of protein, at least one protein, that prevents this aggregation, and the parallel is the chaperone system that prevents the aggregation of the other proteins, right? So at the same time, these show us one, and there is other examples of, it seems for me is the opposite. Is the system was designed to aggregate, and it, in my impression on the field, I'm not in the field, but my impression was also that those kind of mRNA granules, they have some purpose. It's not clear, but I was shocked by your slide.
So
that part of this is garbage.
good. So okay. So you were paying attention. So that's good. So I think the it's a continuum. So let's think about protein assembly as because something we're all familiar with. There are certainly large protein assemblies that cells make using specific interactions between proteins to build those things, right? And there are also cases where cells can make large aberrant protein assemblies through non-specific promiscuous interactions, right? Which will give you protein aggregates and toxicity in some cases. I think we should think about RNA the same way. There are going to be cases where RNA molecules, evolution has selected for specific interactions of the RNA or the RNP to form structures like what Liz is discussing that have defined biological functions. And that evolution has worked on those to do so in a way that's interesting and imparts some specificity. I think there are other cases where, I think particularly a stress response where you're releasing, a huge amount of mRNA from translation very rapidly, where maybe the cell doesn't really, build a specific structure. It just jams everything in a granule because there's an advantage to that probably has to do with signaling pathways that we don't fully understand yet. So I think you're absolutely right, Fernando. But the way to think about it is a continuum. On one end, structures form that have been evolutionarily selected for that take advantage of the biophysical properties of macromolecules and cells. And on the other end, those same properties can cause those proteins and RNAs to form promiscuous non-specific assemblies that can be toxic, and cells have to guard against that. So it's a continuum across that gradient. I hope that answers your question.
I would also add, and this is a point that actually Roy made to me at a conference a while back, was that like the kind of granules that I'm looking at are very, very stable. So these are long-lived granules. They're made in the oocyte. They have to maintain those RNAs intact for hours and hours, many hours, in fact, days in some cases. Whereas stress granules, P-bodies, these are transient structures, right? And they have to be able to form and unform in fairly rapid time. So I think, again, there's also this idea of, in some cases, you've got these granules that have to essentially turn into rocks, and ultimately be very, very solid. And we have evidence that the RNAs are not moving. There's not a lot of fluidity in the germ granules, which is different from, at least our germ granules, which is different from things like stress granules and P-bodies. So I think, again, that just another sort of component to it is the sort of the long-lived versus transient nature of these structures.
Okay, thank you. More question, please, Tatiana.
Well, I was curious when
there is a question from
okay.
Deborah. Could you read, please?
Oh, Deborah can actually, if you want to ask her directly, Deborah, go ahead. Well,
so I could read Deborah's question if that's helpful. So Deborah actually is asking, which is a really interesting question. So in tau aggregation, I talked about this SC35 epitope. The protein that it recognizes is referred to as SRM2. so it moves from the nucleus to the cytosol. What are the disease manifestations of this that could be explained by a shortage of the SC35 epitope, SRM2, in the nucleus? Okay, so this is a really interesting question. So SRM2, which is the protein tagged by this antibody, is a protein that has a role in modulating alternative splicing pathways. And so what we see in cells where that protein has mislocated to the cytosol is that we see a decrease in overall splicing, and we also see certain specific alternative splicing pathways that are changed. It's very well known that in tauopathies and in Alzheimer's disease, there's tremendous amounts of splicing changes in the brain of biopsies. We can't say that, oh, look, we can explain all those changes by this one protein moving to the cytosol. And we think we can't explain it because of two reasons. One, it's not just that one protein. I went over this pretty fast, but there's several other proteins that move with it. So it's not a single protein that's mislocalizing, it's a number of different ones. And the second is that when you sequence a biopsy from a patient brain after they passed away, it's a very complex mixture of different cell types and different stages of the disease. So it's really hard to relate that back to what we're seeing in a cell line model or even in a mouse where it's more controlled. So, but I think the big answer to your question, Deborah, is that there's going to be changes in RNA processing because of this mislocalization. And then the question is, how much of the disease is caused by that mislocalization, and how much of the disease is caused by other aspects triggered by tau aggregation? And we just don't understand those things in these diseases yet. And but at least I think we have some hypotheses we can test now. So if I can ask a question, so Liz, how do you think about specificity then? If there's, molecularly, what does this mean in terms of interactions between the MR and Ps?
You're muted.
You sound, yes.
Sorry. I always try to keep myself off. There are, yeah, it's a good question, and it's something we haven't obviously figured it out. But clearly there's some feature that the RNA can tell itself from a different RNA. So there's got to be some kind of RNA, I presume, RNA interactions or RNA protein interactions, but they can't be, they have to somehow have kind of cover the whole gamut of the RNA, or they have to have it has to be some sort of more holistic property of the RNA. And, so it can't be just, like the way Oscar dimerization through two loops. It's got to be something much more complicated than that. And so I assume it must be structural in some way, but exactly how, I don't know.
So but is it a fair inference from your experiments that GFP has this property as well?
Yeah, it has to, right? Because if you get GFP into a granule, it can actually it'll form homotypic clusters of something with, of GFP. So I think the reason it forms the clusters in the granules is the concentration is just, super, super high, right? But that still doesn't mean that, it's so, it can find itself and but what biophysical property or what specific interactions are mediating, GFP being able to see GFP? I don't know. I don't know. I mean, I don't know how to think about it. If you have ideas, I'd love to hear them.
I mean, the simple model would be that the Drosophila RNAs have been genetically selected to have this property.
True.
But the fact that GFP does it as well kind of makes you, wonder what the hell's going on. So it's really interesting. I don't have a good answer.
Yeah.
So Liz, do you know what prevents the mRNAs from aggregating when they are circling inside the zygote?
Right. So my guess is it's, they need to be at very high concentration in order to undergo whatever kind of self-recognition and phase, some kind of phase separation that there, or condensation reaction that's causing them to go into these clusters. So my guess is that they're just not at high enough there are reasonably high concentration in the cytoplasm, but they're not they're not close enough to each other and high enough a concentration to actually do that. So I think it's probably a concentration. The other possibility is that there are helicases associated with these granules. And so it's possible that, the helicases need to unwind or expose parts of the RNAs that are going to mediate whatever it is, however it is that they self-recognize, that the helicases in the granules are required to unwind them or expose these epitopes on the RNAs that actually mediate the self-recognition. And it could be both.
Depletes the ATP.
Yeah. It'd be nice to try that.
Okay, thank you guys. Thanks a lot. There is the last question is from Jess. Could you read, please, Tatiana?
Sure. So there is a recent report from Kayed's group that demonstrate co-aggregation of tau with p53. They both tend to aggregate. RNA could act as a modulator of gain of function in AD.
So that's that's an interesting observation. I'm not familiar with that paper. It's certainly clear that cells with tau aggregates do induce p53, probably because they have DNA damage being triggered. But I'll have to look at that. That's the co-aggregation would be actually really interesting, and I'm trying to think if we've seen that in any of our mass spec data, but I don't remember. But I'll take a look. Thanks for bringing that up, Jerson.
There's another one from Deborah. Do you still have time, Fernando?
The last one, please.
So what happens with the zygotic mRNA expression if Oscar does not go down?
So zygotic RNA expression in the germ cells? Is that the question? Or the MZT in general?
Yeah, she does not specify.
So, we haven't looked at,
zygotic mRNA
right. So nothing, if Oscar doesn't get degraded, it won't affect the maternal zygotic transition of the degradation of the broad scale degradation of maternal RNAs. What we haven't looked at is whether it affects the so ultimately the germ cells themselves, the pulse cells, will undergo their own maternal zygotic transition. And it's possible that it's somehow screwing up that later, but we have not looked, so I don't know. But the main maternal zygotic transition that happens within the somatic portion of the embryo will proceed normally, regardless of whether or not Oscar gets degraded.
Okay, thank you very much, Dr. Roy Parker, Dr. Liz Gavis, for very, very nice talk, discussion, and everyone for the questions. I hope to see you guys one day in the RNA conference.
Yeah, good seeing you all. Thank you, and good seeing you, Liz.
Yes,
Take care, everybody.
Thanks, everyone.
Bye-bye.